to cloud. Okay, good. Good morning, good afternoon, depending where you are. Thank you very much all for coming on this Friday. This is the first time we switch IVE meetings to Friday. We're going to do that for the rest of this semester at least. Today, it's my great pleasure to introduce Xiaowei from University of Pittsburgh, the Saxena Research Group. As many of you already know, that group is working on double histidine copper binding as a spin probe to study structural dynamics of metalloproteins, right? So today, Xiaowei will tell us something on force field, newly developed double histidine copper force fields, identify cost-efficient DHIR collection protocol at Q band. So with that, I will handle the screen to Xiaowei, and then thank you again for telling us your story.
All right, thank you, Yan, for introducing me. Today, I want to tell you the story of force field development for this double histidine copper motif and how those force fields help us identify the most efficient way to perform Q-band DHIR. All right, so first of all, what is dHis copper, right? So the Saxena group has developed this dHis copper motif that anchors copper in between two carefully positioned histidine residues. So this kind of spin label doesn't have flexible side chains, so it's highly rigid. If we introduce two spin labels onto a protein, we can then acquire EPR distance measurement between the spin label sites, such as DHIR measurements, as we might be familiar with, right? So this dHis copper labels results in 5 times narrower distance distribution compared to the commonly used nitroxide spin labels, which means that dHis copper can better resolve protein backbone motions. So recent applications shows the power and potential of these spin labels, such as characterizing multiple functional states that a protein can exist in one sample. We can see this distance distribution is highly resolved, and each of the distribution can relate to a different conformation, which cannot be resolved with nitroxides. All right. Further, using a minimum of only 4 distance measurements, Bogetti et al. Were able to find the exact location of the metal binding sites. Last, using the orientational information, this spin label can behave at high frequency. Bogetti et al. Were able to figure out the relative angles between 2 protein subunits, which adds in dimensions of the protein structure and information. All right, after we obtain all those EPR distances, we often want to relate EPR distance to computer models. And nowadays, molecular dynamic simulations are a powerful tool to do so. Right, so first, the first part of this talk, I will introduce you the force field development for this dHis copper label so that we can use these labels within the protein and simulate the spin labeled protein. Next, using the key information from the MD simulations, we were able to explain the orientational selectivity effects that happens in X-band, and also using those simulations and experimental validations, we figured out the most efficient way to perform Q-band DHIR using such spin labels. First of all, I'm showing you a workflow, how we derive the force field parameter. This piece of work is done in collaboration with Dr. Jimmy Wang, who is part of the AMBER community, and he's also located in University of Pittsburgh. So first of all, we derive the van der Waals parameters for copper. Next, using DFT, we obtain the optimized 3D structure of the spin label, and there are 2 spin labels we use in our lab, dHis copper NTA shown on the left, and dHis copper IDA shown on the right. Right, so here shows the 3D optimized structure. After we obtain those structures, we can then derive the partial atomic charges for each of the atoms involved in this spin labels. And last step is to derive the bonding terms that copper is involved. And for the other terms, we can just use the existing amber force field parameters to assign. All right, after we obtain all those force field, we want to validate our force field with experiments. So here we performed MD simulation on this 4 different label systems showing at this left corner of each figure, right? And then we obtain the MD sample distances showing this black curve and compared to the EPR distance showing in this blue dashed curve. And we can see the MD sample distance distribution agree very well with the EPR distance for all 4 cases. And given how narrow the distances are, we can still get the MD distance spot on the EPR distance, meaning our force field is valid and is capturing what we should be seeing with the EPR experiments. Another way to validate the force field is to see introducing copper labels to the protein, will the protein dynamics be influenced? Right, so here I plotted the C alpha distance distribution between the labeled sites with and without the spin labels. In the blue is the with the labels, and the black dotted line is without labels. We can see the C alpha at the label sites aren't influenced by introducing the copper labels, and that means our force field is good, is valid, is compatible with current protein force fields. So another thing we want to explain beyond distances is the orientational selectivity effect. Let's say we perform DHIR measurement at different magnetic field G parallel here, right? So we expect to excite molecular orientation in this particular manner with theta value around 90 degrees in this system. The theta is the interspin vector with respect to applied magnetic field here. And let's say we perform DHIR at this G perpendicular field, we expect to excite a very different molecular orientation. And this theta should be sampled around 20 degrees. And we know DHIR measurement is not just related to the distance between the label sites, but also the molecular orientation, theta. So in 2 different fields, we should be seeing very different modulations in DHIR measurement. However, no matter where we place our measurement at across magnetic field, we always see the same results. How do we explain this phenomenon? In reality and in simulation, we know that copper exists in very elastic coordination environment, which means we can sample a range of bond angles and bond lengths. So what we did was we extract many snapshots from the MD simulation, hundreds of snapshots, and use DFT calculation to analyze the G tensor orientation of 2 spin label sites, spin A and spin B, and plot G tensor orientation for both spins. And we can see the blue sticks is occupying a lot of areas in this 3D space, meaning our G tensor is, taking a lot of orientations within the trajectory. Let's take a look back to excitation at a G parallel field. And we just see one snapshot where spin A and spin B points at this particular directions. Right now we are aligning the G parallel with spin A to the applied magnetic field, and we can excite molecular orientation in this way. Let's take a look at another snapshot where spin A and spin B points at very different directions. Right now, again, we are aligning the G parallel with spin A to the applied magnetic field, and the molecular orientation excited at this particular manner is very different. Let's take in all the hundreds of snapshots into account. Again, aligning the G parallel with spin A and see where spin B is at. And we can see spin B is occupying a lot of space indicated by those orange spheres, which is kind of like a meatball you are cooking in your pot, right? So that explains why at a single magnetic field, we can excite so many molecular orientations. So X-band doesn't show any orientational selectivity using this particular spin label. However, if you want to increase your sensitivity of your DIR measurements, we often want to use higher frequency to perform this DIR measurements. However, for the dHis copper labels, it shows highly orientational selective, meaning if you perform DIR across a magnetic field, you get very different modulations. And Bogetti summed up 17 different DIR measurements across a lot of magnetic field to have an orientational independent DIR measurement that you can trust the distances. However, the question is, do we really need 17 measurements? And what is the optimal way or optimal fields that we should perform DIR? So Zikri Hasbasri, who is my lab member, we have been working to develop this protocol to efficiently collect Q-band DIR. So first we did was we created an in silico model that contains 10,000 spin pairs. We just ignore all those protein structures and where we put the spin labels are. We only focus on the 2 spins. So this spin pairs includes a spin A and spin B connected by an interspin vector R. And this R is sampled in the MD simulation. It's a pretty Gaussian shaped distribution. And our goal in the sampling is to properly sample theta that I just talked about in the previous slides, right? We want to properly sample the molecular orientation in our DIR measurements. However, this theta sampling is highly dependent on how your spin A and spin B oriented with each other. So it's system dependent, right? So we want to look at a more general term that we can look into and doesn't have all those effects relating to the spin A, spin B orientations. So we instead looked into this spin orientation called phi. And the spin orientation is the angle between the G parallel and applied magnetic field, right? So in theory, if we can properly sample the spin orientation, we should be able to properly sample the molecular orientation. So this is the goal, right? And now we then quantify the number of phi that can be excited at each magnetic field, shown as a phi curve in this black dashed line over here, and find the maximum, which corresponds to the field that can best sample spin orientations. But the question is, will one measurement be enough? Will this single magnetic field sample enough molecular orientations, right? So now we have to go look back to the theta sampling at this magnetic field and determine if it is good enough, right? So in the ideal case, the theta sampling should be around a sinusoidal curve in this black dashed line. But we can see at this single magnetic field, we are only sampling this much, and there is undersampling between 30 degree to 90 degree. And this means we need to probe the spin pairs somewhere else. So what we did was we subtract all the spin pairs that can be excited at this magnetic field, use the leftover spin to rebuild this phi curve, and find the maximum again. So this is the second field we want to theoretically perform our DIR at. So after we sample spins at this 2 particular field, we can see a slight increase in the sampling between 30 to 90 degree, but not quite there yet. But after including the third magnetic field, doing the same way we did with the second field, we can see a converge in the theta sampling. So theoretically speaking, 3 fields should be properly sample our molecular orientations. Now we want to see the simulated DIR with excitation at 3 magnetic field versus the ideal case with all the spins excited, right? So excitation at 3 fields, DIR time trace is shown as this blue line, And this black dashed line is showing the full excitation and shows very good agreement between the two. However, this sampling is based on, we know the spin A, spin B's orientation with respect to each other from the MD trajectory, defined by chi, gamma, and eta. But in the unknown case scenario that we don't really know the protein structures, how the spin A and spin B oriented with respect to each other, how do we know if the D are going to work at 3 fields, right? So we perform this simulation in combination with all the possible chi, gamma, and eta values, and we can see very good agreement, 3 fields versus everything excited. So in theory, our idea should work. And now we want to experimentally evaluate if this 3 fields methods is going to work, right? So we performed DR measurements on this model system called glutathione S transfers, and its ligand bound form has already been solved, showing the PDB over here. So since it's a homodimer, right, there is a monomer here. So if we introduce a single dHis labeled sites onto one monomer, we end up with 2 labeled sites, and we can perform DR onto this protein. So we perform DR measurement at these 3 specific magnetic fields, right, suggested by the previous theoretical calculations, and we can see the 3 DR measurements shows very, very different modulations, meaning this system is highly orientation selective, right? After we have reached the 3 DRs all together, and the average DR time trace shows up here, and a result in a distance distribution centered around 5.3 nanometer with a standard deviation of 0.2 nanometer. So we want to see, after including more and more measurements, will the DR time trace change? So we perform 7 additional DR in between magnetic fields, and the 10 fields DR looks exactly the same as the 3 field in terms of modulations, and how deep the signal is modulating. And so does the distance distribution. They agree very well. Again, the MD sampled distance distribution shown up here in this blue dashed line, again, agrees very well with the EPR measurements. And this piece of results is in growing evidence that MD can accurately report EPR distances. And last, we want to further validate our three field measurements using Q-band DR, using X-band measurements to further validate our Q-band DR. We know from my previous slide that X-band measurements using dHis Cu2 isn't orientational selective, so one distance measurement is enough, right? So we can see the resulting distance distribution for the Q-band DR versus X-band DR shows excellent agreement again with the most probable I have labeled with the red dashed line. All right, hopefully in this talk I have covered, for the, general workflow for force field development for dHis Cu2, NTA, and IDA. So if you are interested in some other spin labels you want to develop force field with, you can refer to the protocol that we published in our paper. And using those parameters, we have seen very good agreement between the EPR distance and MD distance. And also using the key insights from the simulation, we were able to explain using molecular basis that why X-band DR doesn't show orientational selectivity. And a strategic excitation of only 10% of the copper spectrum is enough to properly sample all molecular orientations at Q-band. And this 3 field measurements reduces the data collection time by at least 6-fold. And this 6-fold is very significant thinking about if you are using helium all the time, right? And this we have published this Python code to calculate the optimal fields. If you are interested in some other orientational selective pulse EPR techniques and other paramagnetic ions, you can check out our code to adapt it. And last, there are two key publications that are related to today's talk, and if you're interested, please check it out. And I will thank everyone for listening to me today. And I know it's a Friday, but thank you all for coming. And I will thank Saxena Group for all the supports. I will thank my collaborator, the funding, and PCRC to carry out my simulations. And again, the key publication related to today's talk is attached here again. All right, thank you. I will be glad to take any questions from the audience if there is any. Oh, I think there is a question.
Hello. Yep. My name is Graham Smith from St Andrews. So very, very nice talk. I enjoyed that lots. So a long time ago, we also did similar things on nitroxides, looking at orientation.
Yes.
And if I'm remembering rightly, I mean, our conclusion was because in your measurement that I guess you're just correlating two parallel orientations at three different fields. And I guess my question is that really sufficient? I always thought you needed to also correlate orientations at different field positions. In other words, do your deer experiment pumping and probing at your three different points. But are you saying then that effectively that's not required? If you understand my question. Like for example, supposing your spins were at 90 degrees to one another in terms of their orientation, then you'd expect to have the biggest modulation by measuring pump and probe far apart. But are you saying then that actually you don't need to make those measurements, that the three measurements that you're doing just by measuring the parallel orientations is sufficient, and there's no ambiguity there?
I guess I kind of understand your concern here. So just because at a single magnetic field, right, you can correspond to many different spin orientations actually because of the G and A and isotropy. So you are not just exciting a single G parallel, but it's a range of, G orientations and A in the single field. So even at the higher field, you can still excite spins where you are exciting some of the 90 degree, which is a G perpendicular area.
Yeah.
Yes.
I see what you mean. Okay. Perhaps an analogous question is does the method assume that there's a single orientation, or can you work out the distribution of spins in the octant?
I'm sorry, can you repeat your question again?
Are you assuming that there's a single orientation, or can you work out what the distribution of orientations is?
Yes, it's a great question. Actually, so when we are generating the spin pairs, we actually randomly rotating the 3D field, so it's covering a sphere of different orientations. So the spin doesn't really only point at a single direction, if that answers your question.
I guess the question is, can you quantify that?
The question is, if at a single magnetic field, if we can quantify the number of spin orientations, correct?
Yeah, or can you quantify the distribution of, whether it's a single orientation in the ideal case, or whether it's I guess if it covers more than an octant, then you just see the average all the time.
Yes, and so I think that the phi curve sampling Let me just go back to a few more slides. Sorry. Yes, so the phi curve sampling. So we also plotted, the range of phi that can be sampled at different magnetic field. It actually samples a lot of molecular orientation, the spin orientations actually. So we can excite a range of spin orientations in the 3D space. If that's a better answer.
Okay, but I was really asking, can you quantify that though? Do you end up with a distribution, or I'm don't doubting you're exciting them, but can you quantify it?
Yes, we can quantify it. So we can count at each, the phi value, how many we are exciting. Yes, we do.
Okay, thanks very much. I'll let somebody else.
Thank you for
very
the
nice
question.
talk though. Enjoyed it.
And I think Sang have a question.
Hassan, just ask your question.
Yeah.
Unmute yourself.
Okay, yeah, that's fine. Very, very nice talk. I do have very quick questions. I didn't quite understand the setting of this three-field position. So what offset you were using between pump and observer pulses, and are you setting this three-field position and the pump is fixed, or yeah, if you can a little bit give details about this, that would be great.
Yes.
Okay, thank you.
It's a great question. So those three fields, actually we put the pump pulses at those fields so that we can excite the most spins that gives us the best modulation depth at that field. And the observer pulses are placed around 100 megahertz away from the pump.
For each field position, you have 100 megahertz offset.
Yes, exactly.
Okay.
And
yeah.
This theoretical calculation is just assume the pump and observer always 100 megahertz away, which probably can work with most of the commercially available resonators. But if you have a better bandwidth resonator, let's say you have 400 megahertz or 800 megahertz, you might need less and less measurements to do.
Okay, thank you.
Yep, no problem.
So, Xiaowei, there is a question from the chat.
Oh,
Is
sorry.
that three-field Q band sufficient?
Was the distance distribution drastically different if you used only one or two fields? So yeah, it's a great question again. If we only perform deer at this red field, and I will show you an example what the deer looks like, right? We can see this red field is corresponding to this purple field that I collected deer, right? Then you're gonna resolve in a very, very sharp short distance. We can see this small modulation over here. This is due to an orientational effect rather than a real thing, as we can see in the final distance distribution. And even you only include two fields, you still have a very sharp feature around some shorter distances in this case. Yes.
So basically multiple time traces are needed, right?
Yes.
Can you suggest us what's the minimum number of traces that are needed?
Actually,
the more the better.
If you have a ultra wide bandwidth spectrometer, let's say you can excite a gigahertz,
right.
You only need one.
Oh, okay.
Based on our theoretical calculation.
Oh.
And if I know different lab has different resonators, and if your lab have a different bandwidth or a larger bandwidth, you can use our code to calculate how many fields you really need and see if that's gonna work. Yes.
Okay. Cool. Any other questions about Xiaowei? No. Okay. Well, if no more question, well, let's thank Xiaowei one more time. Great talk.
Thank
really
you for
try it.
this opportunity.
All right. We'll see everybody in 2 weeks, I guess. Thank you.
Thank you, guys.